Alright, Chapter 27, Part 2. So now we're going to go into the actual production of sperm and going through the process called meiosis, where our initial parent sperm will eventually, parent cell will eventually develop into the different gametes. So the process of actually producing sperm is called spermatogenesis. It occurs in the seminiferous tubules, which are located in the testes. So there's 3 events to produce sperm. It's the division and remodeling of the large germ cells into the smaller sperm cells with flagella. So that's the flat whiplash tail at the end. The chromosome number is reduced by half. So you're going to go from 46 single-stranded chromosomes in a normal cell down to 23 single-stranded chromosomes in a gamete cell. During this process, the genes are shuffled around, so you get new combinations of genes that weren't in the parent, and maybe, hopefully, with the combination with the other parent, then the offspring has a better chance of surviving, hopefully. So this process to produce the 4 daughter cells that will become the gametes, in our case focusing on the male reproductive system, those gametes being sperm, is called meiosis. So both mitosis and meiosis are some type of cell division. So there's a couple slides which I'll have to, I'll kind of break stuff down a little bit more individually than the way the book does. So some of these figures aren't located in the book. They're just stuff I made for the PowerPoint. Okay, so we've gone through mitosis from last semester. Its job is to produce genetically identical cells. So just your regular normal cells creating more cells. So you start off with your one parent cell that has 46 single-stranded chromosomes. This is called a diploid cell, so sometimes you'll see it marked off as a 2N cell. This one parent cell will go through mitosis to produce 2 new daughter cells. Each of them are going to have 46 chromosomes, which are all single-stranded, and these newly created cells are also diploid cells, just like the one parent cell, again with the 2N denotation. And these 2 daughter cells are genetically identical to the parent cell. Meiosis, on the other hand, produces genetically different cells. So the DNA is going to be different from the parent cell. So again, another reason why the sperm have to be protected from the immune system, because of they are genetically different. They have half the DNA of a normal cell. So to prevent them from being attacked from the immune system, they are protected by those tight junctions from the nurse cells. Okay, so genetically different, half the DNA in your gametes. So starting off with our one parent cell with 46 chromosomes. So again, this is a diploid cell, also denoted as 2N cell. So this one parent cell will go through meiosis, and we'll go through those details later, to produce the gametes. Gamete cells have 23 chromosomes, so 46 single-stranded here, 23 single-stranded chromosomes here. So half the DNA from the one parent cell in our gamete cells. These cells are now called haploid cells because they have half the DNA. So the denotation for them is 1N. So go from 2N to 1N, go from 46 chromosomes to 23. So being a diploid cell to a haploid cell. And I have the star beside the gamete name because your gamete number depends on the sex as well as reproductive activity. So in males, this will normally, the process of meiosis will normally just produce 4 sperm. However, when we go through the female reproductive system chapter, there are some differences in the gamete production that's necessary for creation of the egg or the oocyte, and so you don't produce your 4 gametes in that condition, as well as I state here, reproductive activity. So there's the process of fertilization. So there's your sex reproductive activity, where that will finalize your meiosis to finish up the final steps. Okay, so as you're looking at your different terminology, unfortunately, when the chromosomes were named, whether or not they were single-stranded or double-stranded, they still have the title of being a chromosome. So a chromosome could be a single-stranded, unpaired strand of DNA or a double-stranded pairing of DNA. So it'd be both copies of the same set of DNA and they're paired together, and that's still called a chromosome. So some people find that a little bit confusing. So I'm just pointing this out, even though it could be single-stranded chromosome or double-stranded chromosome. So I'll use this backslash or forward slash, I can't remember. That denotation for a single-stranded one, the X for a double-stranded. So these are both copies being one from one parent and one from another. Okay, so here's where it gets a little iffy. So you could have a diploid cell with 46 chromosomes that are single-stranded, so unpaired, having the same amount of genetic material as a diploid cell with 23 chromosomes, but has the same amount of DNA or genetic material in it because those 23 are in pairs. And so 23 times 2 is 46. So that's something you just have to pay attention to, whether or not if you're looking at your DNA, whether or not it's single-stranded or double-stranded. Okay, so there's two types of cell division. I'll quickly go over mitosis again and the numbering of the chromosomes. So again, mitosis produces two genetically identical daughter cells. So I'll go to my little flow chart here. Again, this chart is not in your book. So we start off here with our one parent cell, which you guys can't see. So starting with one parent cell that has 46 single-stranded chromosomes, during S phase, that DNA is then doubled. So now you have 92 chromosomes that are single-stranded. Then in mitosis, so the first stage of mitosis, you have your prophase, and then it goes to the metaphase, anaphase, and telophase, which you've learned last semester. So during prophase, this DNA is compacted. So your pairs of DNA are compacted together to form two sets of the 23 double-stranded chromosomes. So two sets. So you have your 23 double-stranded, so that would equal 46 single-stranded. There's two sets of them, so that would equal your 92 single-stranded, except for everything's all compacted together and partnered together. So you end up with two sets of the 23 double-stranded chromosomes in your cell during prophase. Oops. Okay. So after prophase, you have your metaphase, your anaphase, and your telophase to separate all your stuff. Final step, cytokinesis, to split the two cells into the two daughter cells that again are genetically identical because this is mitosis. So each daughter cell after cytokinesis is going to have one set of the 23 double-stranded chromosomes, and then after the final process of cytokinesis, those 23 chromosomes are then unpacked, so uncompacted like from earlier, into the 46 single-stranded chromosomes, just like the one parent cell. So we start off with a diploid cell in mitosis. We end up with two diploid cells in at the end of mitosis. So for meiosis, in the production of your gamete or sex cells, arranged here, there we go. So with meiosis, you have two sets of going through the processes like mitosis. So on the side here, I have your prophase, your metaphase, your anaphase, and your telophase. So that's set number 1. And then you have a prophase 2, a metaphase 2, an anaphase 2, and a telophase 2 to eventually go from one parent cell all the way down into your 4 gamete cells. So the reason for ending up with gametes that have 23 chromosomes, but now they're unpaired, is so that when the two gametes meet during fertilization, 23 unpaired from one parent, 23 unpaired from the other parent, combines to have your original 46 single-stranded chromosomes inside the zygote, and that way it keeps the same number of chromosomes as the parents. It also prevents your DNA from doubling every time you have some kind of sexual reproduction. So it keeps your DNA level at the same no matter how many times you reproduce. Okay, so for your meiosis, you guys can see this well enough, I think. You start off with your one parent cell, again, diploid, 46 single strands of chromosomes. Again, it's a 2N cell, and you see the pictures. This DNA is doubled before going through our mitosis-like steps. So we go from 46 single-stranded chromosomes to 92 single-stranded chromosomes. Then we go through our mitosis-like steps, so our prophase, metaphase, anaphase, telophase 1. So during prophase 1, these 92 single-stranded chromosomes will compact into 46 double-stranded chromosomes. And later I'll show you a picture where as these chromosomes line up with each other, they end up forming new gene combinations, and this is how you end up with gametes that are genetically different than your original parent cell. So after going through the mitosis-like steps, splitting up at the end of cytokinesis, ending up with one daughter cell that is diploid, so same as up here, and your chromosomes are double-stranded, so they're paired up, so 23 chromosomes there. So if we were to split these apart, they'd still be 46 chromosomes inside that cell. So we have two cells here. Then for the next step in meiosis, it goes through those mitosis-like steps again. So prophase 2, metaphase 2, anaphase 2, telophase 2, ending up in cytokinesis, where you produce your 4 gamete cells. Again, this depends on whether or not you're in the female or male reproductive system, but since we're talking about the male one, we make 4 gametes, so our 4 sperm cells. So with our 4 sperm cells, or our 4 gametes, these cells are haploid. So these 23 double-stranded chromosomes are split apart, and now each of the gametes have one strand of that 23 paired. So these haploid cells only have 23 single-stranded chromosomes. So they have half the DNA as your parent cell that used to have 46 single strands, and now splitting that in half after your whole process here, down to 23 single-stranded chromosomes, which is a haploid cell. Yay, okay. So next slides go through it in a kind of little bit different manner, just gives you more detail. So again, your meiosis has 2 cell divisions, so our 2 mitosis-like steps, still having your prophase, metaphase, anaphase, and telophase. So in our meiosis 1, you have the 1 designating that it's in that first set. So you have your prophase, where that nuclear envelope disintegrates, those DNA are compacted, and in this phase, your chromosomes, as they line up side by side, so this chromosome is, so you have DNA is doubled, and then this chromosome is lined up with its partner chromosome that is double-stranded, and sometimes when they're in this tetrad, so 1, 2, 3, 4 strands of DNA, so tetra meaning 4, sometimes they'll cross over with their DNA and so make different combinations. So during metaphase, we see here there's been some switching of some genes. So now this mostly blue chromosome has a little bit of from the red chromosome, and this mostly red chromosome has a little bit from the blue chromosome. So that's where you get your different gene combinations, and hopefully your offspring that is possibly produced from these gametes will have a better chance at survival because they're not exactly the same as the parent. Okay, so next few stages is kind of normal, like your mitosis, so your anaphase where everything gets split up. So grabbing those chromosomes, splitting them from their copy set into your two daughter cells. So these two daughter cells have the same amount of DNA as the original parent cell because the DNA was doubled before going through these meiosis 1. So now going on to meiosis 2, again having a prophase, metaphase, anaphase, and telophase. So now we have the 2 designation. So instead of your DNA unpacking and going into single strand, it stays into its compact stage. Prophase, again, that nuclear envelope disintegrates. Metaphase, where it aligns on the center. And then this stage, from the centromere, they actually splits the chromosomes apart. So going from double-stranded chromosomes to single-stranded chromosomes, splitting the genetic material into, so you have your 2 cells, splitting that genetic materials into 2 cells themselves, making your 4 gametes. Okay, so this picture has everything in one page. So this side being meiosis 1, so DNA doubled. Then you have that prophase 1 where there's some gene switching, so get some new genetic combinations. Then that double DNA is split in between 2 cells. These 2 cells still have the same amount of genetic material as the parent cell, so still a diploid cell. Then these 2 diploid cells up here start going through meiosis 2, which is second phase here, where the nuclear envelope disintegrates again, then going through that whole process where instead of the DNA doubling, the DNA is now split. So splitting up these double-stranded chromosomes that were 23 double-stranded pairs. Those 23 pairs are essentially ripped in half. So each copy of one of those double strands is sent to a new cell. So you have your 2 daughter cells that were left over from meiosis 1, now splitting into 2 cells to make our 4 gametes. Okay, so focusing just on sperm development, down here we have our spermatogonium, which are our parent cells. So here the 2N designation being a diploid cell. So of these spermatogonium, the type B is the one that will eventually become sperm. So our type B spermatogonium will go through meiosis 1 to split into a secondary spermatocyte. Then this secondary spermatocyte will then split again through meiosis 2 to form your 4 haploid cells. So here they're showing the 1N designation, but it still has the same amount of genetic material as this cell. It's just that because those are double-stranded, so it's like 46 here, but then it's 23 here. They give it the N, even though here that haploid cell here has half the DNA as this one. But keeping your head straight, DNA is doubled. That double DNA is split into these spermatocytes, which have the same amount of DNA as the original cell. Then that same amount of DNA is split in half into your spermatids, which will eventually mature into sperm. So here, this kind of orange area is that open lumen in the seminiferous tube where those tails are sticking out, and I'm going to show you a picture of that in a second. So here's our seminiferous tubule. Then this little triangle here is where the interstitial cells where the testosterone is created. And this open area here in the center is that lumen where those sperm tails are sticking out. So those sperm tails will then float along through the fluid that is made by those nurse cells, and then if they are ejaculated, becoming part of semen to exit out of the male reproductive system. So after we've gone through the two stages of meiosis, we have our primary spermatocyte, which is now genetically different because it has half the DNA as a normal cell. So it must be protected from the immune system. So as that cell moves toward that open lumen of the seminiferous tubules, new tight junctions form between the nurse cells behind it to protect it from the immune system. So again, now protected by the blood-testes barrier made by those tight junctions between the nurse cells. So this obviously doesn't look like it's going to do too well with swimming. Not very... Oh, I've forgotten that word. Not very fluid dynamic in its shape. So there's a process to help develop this, give me a second, sorry, spermatocyte into the mature sperm. So through these processes, there's a little bit of flagellum at this stage, and then your cell starts rearranging some of its parts, getting rid of excess cytoplasm, so going from this big fluffy cell to a nice, more smooth, more fluid dynamic, aerodynamic, that's what I meant to say, aerodynamic type of cell, makes it easier to swim. So parts I'm going to point out, so here we have our acrosome, which goes towards the tip of the sperm head. That is filled with different enzymes to help break apart that external barrier on the egg so that the sperm can get inside and fertilize it. The mitochondria, which, like I mentioned earlier, might not remember, but well, so these very large mitochondria that make the sperm able to not only survive the low oxygen environment inside the testes, but also the low oxygen environment in the vagina so that it can survive for a few days until it hopefully can fertilize the egg. So those mitochondria are moved into the mid-piece of the tail, and then as more and more excess cytoplasm is shed off, makes a elongated tail, which allows it to have a good propulsion system so that it can swim and hopefully make it to the egg. Okay, so quickly the parts of the sperm. So here at the top you have the head, again with the acrosome with those enzymes to help penetrate the egg. In this section here is where your genetic material is. So once that sticks through, unload the genetic material and let the baby making begin. Then this middle piece where those mitochondria are to make your ATP energy to fuel this last end part of the tail where the flagellum is, so that it has the energy to cause the flagella to move and therefore able the sperm to swim. So here's the full mature spermatozoan, acrosome tip here, head here, so this is actual sperm. Here's our mitochondria, and then the end part of the tail. Okay, so we've made sperm. We also need the fluid that the sperm is going to be delivered into the vagina. So your semen, which is ejaculated, it's about 2 to 5 milliliters of fluid. Again, 60% of it is seminal vesicle fluid, 30% from the prostate, And 10% left. The last part is part sperm and part secretions from the spermatic duct. So a normal sperm count is about 50 to 120 million sperms per milliliter. So let's say even if you're only at this 2 milliliter of ejaculation or ejaculate from the penis, then that's still 100 million sperm cells in that little bit, even not in a low count. So if it's lower than 20 to 25 million per milliliter, the person is considered infertile, just because the environment to get from not only getting out the penis and surviving, but also the vagina and uterus, as well as uterine tube environment, is very anti-sperm. So in order for them to survive all the way to the journey towards the egg, they have to go through an acidic environment inside the vagina. And then there's the mucus that surrounds the cervix. And then they actually have to get into the uterus. And then once they get into the uterus, they get hit with white blood cells. And then there's also the fact that, hey, you're in the uterus, there's a bunch of folds inside of it. And so they get trapped. You know, of course, they didn't ask for directions, which way to go. So this is also why it's kind of a good thing if you're actually trying to have a baby, making sure that your female partner has an orgasm, because it actually pulls not only the semen into her uterus, but also pulls it to the direction. So since you have one egg released, you have 2 possible directions, so it helps to pull it into whichever particular direction towards the egg that was released. Okay, so eventually you get through the uterine tube, which again, that's another hit of immune system protection, and eventually you finally get to the egg, and even then you still have this hard exterior you have to break through. So yeah, even though there's a ton of sperm cells, there's a ton of stuff, this big gauntlet of what's called the female reproductive system to possibly have to survive and be able to fertilize that egg. Okay. So with our semen, again, with our different little parts, so we have our prostate gland, which is right underneath our bladder here. It releases a fluid that contains a clotting enzyme, which will become important in a second, and a protein hydrolyzing enzyme, again, which will become important in a second. The seminal vesicles, which are a little bit further up, those will secrete a fluid with a protein called proseminogelin. So this protein and this clotting enzyme will work together to activating that protein to convert that protein into a sticky fibrin-like material. So what that does, it helps to stick the sperm together so that it sticks to the inner wall of the vagina and the cervix, so the cervix being the opening to the uterus, so that prevents that semen from sliding back out. Also helps to uptake the sperm into the uterus in different clots. So kind of like when a farmer is, picking up hay with a pitchfork, kind of like that, where everything's kind of stuck together. So you get this clumps of sperm being taken into the uterus. After about 20 to 30 minutes after ejaculation, the serine protease from the prostatic fluid helps to break down and liquefy the semen so that that semen can then be able to swim. Hopefully it's been taken into the uterus and give it that free-flowing fluid where it can swim up into the uterus and maybe hopefully fertilize the egg. Okay, so for the sperm to move and get to its hopeful final destination, it has to survive the elevated pH. There needs to be an elevated pH for the sperm to be able to move. So the vagina is acidic. So the glycogen that is in the vagina is fermented, which causes it to have a lowered pH. So the prostatic fluid helps to buffer that acidity, so raising the pH from 3.5 to 7.5, so elevating the pH. The energy sources inside the seminal vesicles, there's fructose and other sugars to help make ATP, so those sugars being used inside the mitochondria to make your ATP. Also, the prostaglandins in the semen help to thin out the mucus on the cervical canal, so thin out that stickiness, which will prevent that, would have normally prevented the semen from going into the uterus. It also may possibly stimulate contractions in the uterus, so not like the contractions like you're having a baby, but in, just drew a blank. Oh, during an orgasm, the uterus actually contracts. So I like contractions, not like, you know, having a baby contractions. So those light contractions help to bring the semen into the uterus, as well as the contractions of the uterine tubes to help spread the semen. So there's kind of like a trap door right at the cervix where it allows those contractions from the uterus to dip into that kind of like cup where the semen can kind of get trapped and help to bring that semen into the uterus, and then the contractions keep pulling on that semen to help it move in the particular direction, a little bit toward, at least towards your egg, not necessarily towards whichever side the egg is. Okay, so that is the end of chapter 27, and we're almost through with AP2 material. Still have the female reproductive system for chapter 28, and then chapter 17, which is actually kind of like a review review chapter, since it goes over hormones, and we've talked about hormones in a lot of our different organ systems. So this last final chapter just kind of puts all the hormones into one chapter and just focuses on them. All right.